This is the people working with me: Jason Working, Danny Glavin, Jamie Elsila mainly. People at the Astrobiology Analytical Lab. And all right, so this is what we do. No, sorry. First, I should repeat what I just told you. For some of you, I guess. I was born in Peru, and I grew up in a small town up until my parents divorced. And yeah, it was really humble origins, I guess. After that, my mom, my sister, and I, we moved to Lima, the capital, where I attended public schools. And it was mostly fun, being bullied. No. And well, I don't know, it's just, I guess, a typical Peruvian person there. When I finished high school, I didn't know what to study. So I told my mom, I don't know what to study. And she said, like, I don't care, you're going to study something. Just make sure that whatever you study can be done anywhere in the world. It can be something that will give you time for yourself, like meaning you don't have to work on the weekends. You can leverage your strengths, which at that time I didn't know which were, but I'm still struggling with that. I guess I'm very patient. So to be a chemist, you need to be extremely patient. And yeah, I'm traveling, I'm enjoying my weekend. So I think those were good suggestions. I got a BS in chemistry. And at that time, there were no PhD programs in Peru. And I really wanted to get a PhD because I thought, I didn't really know why, but I really wanted to get one of those. And so I traveled to Louisville, Kentucky, where I pursued my PhD in organic chemistry. And I became Jose Aponte PhD. Right? And I stayed there for 5 years, working on methods of isolation for natural products and doing synthesis of natural products and learning a lot about analytical chemistry, mass spec, NMR. And so all of those techniques that I work with allow me to apply for a job at the at Brown University. Oh, I think I need to connect to the Zoom. No, I'm sharing. Why is it not? No. Okay. And so eventually I went to Brown, and I was there for 2.5 years. And that's where I learned about astrochemistry and astrobiology and started working with NASA-related projects. And in 2012, I applied for a NASA postdoctoral fellowship. And ever since, I've been working at NASA Goddard, which is located outside Maryland, outside Washington, D.C. And it's one of, if not the biggest NASA center in the US. There are so many others. Of course, the most popular is Houston because we have a problem. Right? But yeah, if you are ever in the Washington, D.C. area and want to visit my lab, you just could send me an email, I guess. It looks more or less like this. We have a bunch of different instruments. This photo is not really updated, but essentially we do different flavors of chromatography and analytical chemistry. You have a question? No. So we do liquid chromatography and we do gas chromatography, and we analyze the compounds using different sources, different types of mass spectrometry. We also do isotope ratio mass spectrometry, and I'm going to tell you why we do that. So we basically, I don't want to say that I'm an astrobiologist because that's a really strange concept to me still. But I can relate to chemistry and therefore prebiotic chemistry. And so I always tell people that, you know, if I need to explain what I do to my grandmother or to my mom, I would be like, oh yeah, I do what astronomers do, but without telescopes, right? So the camera. Okay. So yeah, but it says that the host doesn't. Okay. The host is not letting me. Yeah. Okay. So yeah, so astronomers use telescopes that are either in the space or on Earth to look at signatures that are present in outer space, right? These spikes that you would see in this figure, they are peaks. They are each of those spikes corresponds to a different molecule, and we kind of call that a chromatogram or a spectrum, right? And so they look into far space and tell us, oh yeah, this peak corresponds to methanol or water or carbon dioxide and so on, right? So those are the basic molecules that are in outer space, which could be collapsed into a disk and eventually a planetary system like ours, which will form, planets, moons, comets, asteroids, stuff like that, and perhaps life. So how do I do this type of analysis without using telescopes? Well, we do analysis. We do laboratory analysis and simulations. We work with, in some cases, we work with what we call interstellar ice analogs. These are ices. These are literally ices that form through the combination of different gases and water, and they are irradiated as if they were in outer space. So in outer space, they receive a lot of UV light, for example, right? And so that UV light is a source of energy, and that source of energy would drive chemistry, and that will produce the formation of biomolecules. Same thing for early life, early Earth. Sorry, we can model experimentally the conditions of the early Earth and make the appropriate, we can put the appropriate energy to the system, and we can generate molecules that could be used for the origins of life. However, we could also do the analysis of meteorites. Why meteorites? Because meteorites have seen the formation of the solar system, yet they remain unaltered by biology and by geology. And so they contain all the ingredients for life. And it's like the real deal, right? You're not simulating anything. Meteorites themselves like track all that. And we will use liquid and chromatographic instruments to separate the molecules. In this case, for example, amino acids. Okay. So as I was telling you, the Earth is very mean, and that's why I don't like it. I hate to be in this planet. And that's why we look outside, we look for meteorites, because they keep the record of what happened during the formation of our solar system and our planet, of course. Meteorites themselves can fall anywhere in the world. For all we know, one can fall right now, and will probably kill the people in the first floor, but we'll be okay. This is an example of a meteorite falling in Costa Rica, in the town of Aguas, Zarcas, which is a type of chondritic material, chondritic meteorite. And there is Mike Palmer, who went to collect some rocks for us. This is literally how we get meteorites for analysis. They will fall somewhere. If they don't fall in the ocean and can be collected, we'll go after them. Well, someone will go and then send it to me. And then, and this is something that we don't do, because the samples, as you can see, are quite contaminated. But some people could buy meteorites, in the market, like eBay, and they're quite expensive. And you can get scam, of course. But anyways, if you're in the search for samples, but the best way is actually asking NASA for samples, because NASA and the NSF used to send people to Antarctica because people want to go to Antarctica. So I don't know why. And walk around the slopes and look for meteorites. Would you like to do that? If you would like to do that, you better sign up right now, because it takes like 18 years to get, there's a huge list of people that want to do the same thing. I hate the cold, so I'm not doing it. But the NSF and NASA would send scientists to pick up rocks. And of course, they train them on how to find meteorites. And they actually do a good job. Like 90% of the rocks that they find in the field are actual meteorites. Don't ask me how. They take them to the Johnson Space Center, classify for the analysis, and so on. And then there is a committee of scientists that meet twice a year. I'm part of that committee, where we receive the proposals of people that want to analyze meteorites, and we give or may not give the samples to them. So again, now you know someone that can give you samples if you have scientific purposes for them. Meteorites are classified very broadly into like stony meteorites and like metal meteorites or iron meteorites. The ones that we care about is the subfamily called chondrites, because they contain chondrules. Okay, I'm not a geologist, I'm not really sure, but apparently these chondrules are really old inserts in the rocks. Those were like older than the sun itself. Like they were like forming before the sun was finishing forming, which is quite old. And among them, we have the carbonaceous subtype. Okay, these are carbonaceous, as you could imagine, are rich in carbon. And why is that important? Because we are made of also carbon, right? They are further subdivided into 8 different subfamilies. But lie number 1, this is all a lie, because like there are like 50,000 meteorites in the catalog, and it's impossible that they all fit into 8 subcategories, right? So these are the ones that we know, okay? So job number 1, there's a lot of work to do on classifying properly the meteorite. But let's move on. The carbonaceous type could have been exposed to really high levels of water towards the lower numbers 1 to 3, or they could have been exposed to high temperatures from the levels 4 to 6. And so as much as they have seen water and high temperatures, their composition was going to be affected, as you could imagine, right? And I want you to bear in mind this, because then we do the analysis of the meteorites, and these conditions will come into play. So just bear in mind that low numbers is high level of water, high numbers is high temperatures. I know you may be thinking, what if a meteorite had a lot of water and also high temperatures? Well, that's not really clear in the system, as you could infer from here. Okay? So let's just make it easy, like high water, high temperatures. Meteorites, like the Murchison meteorite, which fell in Murchison, Australia in 1969, could be, or some carbonaceous chondrite like this one, could be really rich in organic matter. How rich, you may ask? Well, all the ingredients for life, all the stuff that is in a cell, the cell wall is made out of certain molecules, right? The DNA is made out of other stuff, proteins, enzymes are made of amino acids, and so on, right? All of those tiny molecules could be found in the Murchison meteorite. That's fascinating, because all the ingredients for life, that means that were present when the solar system was forming. They kind of survive, and they're like hanging out in outer space in asteroids, right? So there are different ways into people tackle this problem. We have millions of different compounds, and we have like really specific families of compounds that we want to know more about. So I am the one that likes to look at the compounds per family. So for example, I would isolate the compounds, would identify the families, the different classes like carboxylic acids, amino acids, aliphatic amines, nucleobases, and so on. And I will analyze each family and each compound and will quantify their abundance. I will quantify their molecular diversity. But there is people that looks at the whole thing as a bulk, and they say, hey, there is so much carbon, hydrogen, oxygen combined, and there is so much carbon, hydrogen, sulfur combined, and so on. And so it's just, chemists are this crazy. So you pick your whatever you like, but the main message here is that organic matter could be in outer space, and it could be quite complex and abundant. And that is important because there was a period of time in which it was quite of a hell of a time. The Earth, or the proto-Earth, was being bombarded by a lot of asteroids, comets, and that means that a lot of this extraterrestrial material was implanted on the planet, right? And so it is possible that that implantation resulted in the development of polymers and bigger molecules that could eventually have become living organisms. So that's the link between space, asteroids, comets, and meteorites, and the origins of life. And I'm going to give you some more clues of what other clues or what other relationships are present. In order to look at the organics in meteorites, we have to ask ourselves a couple of questions. For example, if we look at amino acids, it's a pity that the thing is there. But anyways, we want to know how these molecules form in the asteroid in the first place, right? And when we work papers and proposals, we ask ourselves what is the implication for these organic compounds. Thank you, Chris. And for example, the fourth solar system formation, meaning where can we find these compounds aside from the Earth? If they are found in asteroids, in meteorites, does it mean that we can find them like in other planets? And if we do, does it mean that there is life, or like it's just abiotic synthesis? So we can also think what happened in the asteroid. The asteroid was like a making machine, making organic makings, entity, and what processes led to the origins of life. And to do that, we measure the isotopic values. I'm going to tell you what's that about, the molecular distribution, and the chirality. If you're not familiar with those concepts, I'm sorry. No, I'm kidding. I'm going to tell you. For example, what is stable isotopes, right? So the atoms, all of them, most of them, I guess, have like their evil twin brother sister, which has an extra neutron, right? And so their mass is bigger. And so you may have heard of carbon 12, carbon 13, carbon 14. And so the 14 is the radioactive crazy sibling that will decay over time and transform into something else. But the 13 is just the chill, heavy brother that is, very stable through time. And so the ratio of the 13 versus 12 could be, it's very stable through time. So it can be measured. It can be taken as a point of reference. And for example, when we, well, not we, hopefully not we, when people do the Olympic Games, right, and they say, oh yeah, doping. There are two types. If someone sniffs cocaine, they will just look for cocaine in the mass spec and was like, yeah, cocaine, get out, right? Easy. But what happens if the person like injected testosterone? You make testosterone in your body, everybody does. So how can you, differentiate? That's when the isotopes come into play, because the testosterone in your body is out of whatever you had for lunch, whereas the testosterone in the injection is whatever the pharmaceutical company somewhere use, to make that. And so the level, the ratio of carbon-12 to carbon-13 is different, and you can definitely differentiate between them, and so that's how you get cheaters. But in our field of study, we can use the isotopes to differentiate molecules from outer space from those on the Earth. In outer space, molecules have not seen biological processes, they have not seen the geological process, and therefore the isotopic signature is different to that on Earth. On Earth, we are enriched on carbon-12 because biology is quite lazy and likes to break the weakest bonds, which is the carbon-12 bonds. Whereas in space, that doesn't happen, and so the amount of carbon-13 has been stable through all this time. And so that's how we can differentiate extraterrestrial compounds from the terrestrial compounds, terrestrial contamination. The other issue that I was going to tell you is molecular distribution and homochirality. Life on Earth is homochiral. We are made of amino acids, and although amino acids could be quite diverse, we only use 20 of them for life, right? There's no more than a pool of 20 different molecules that we use for making all proteins and living organisms. But again, that is not a limitation for prebiotic synthesis, because in space, and they go through mechanisms like radiation chemistry, and you can find all the different isomers, all the different types of molecules that cannot be made through biology. And that's how also you can differentiate between biological processes and space chemistry. Homochirality, for those that are not familiar, it's a quality of life. We don't really know if it's mandatory for life to be like homochiral to exist, but we know that living organisms are mainly made out of left-handed amino acids, and we know that left and right-handed amino acids have the same physical chemical properties, and therefore they should have the same potential to be used as for the ordinance of life. But that didn't happen for whatever reasons that we don't know, although I think I have the answer for that. Life picked left-handed amino acids only. All right, so we get the meteorite from Antarctica. We grind it, we make a powder, we destroy it, and then we put it in a solvent, depending on the type of molecule that we want to look for. In this case, if we want to look for amino acids, we will just put it in water, because amino acids are water soluble, and we will heat them up for 24 hours at 100 degrees, and we will make what we call an extract that looks like that beer ampule there here. And then we will do the chromatography, the mass spectrometry for analysis, identification, quantification, and enantiomeric ratios quantitation. We have analyzed some meteorites, close to 60 now, and we have found something really interesting, which is that, remember when I told you like they could be, the carbonaceous chondrite could be classified as like a lot of water or high temperatures. So it looks like the not too cold, not too hot, not too wet, and not too dry meteorites are the best ones. They contain the largest amount of alpha amino acids, which is the type of amino acids that we are made of. We're made essentially of alpha amino acids all the way, right? And those that have been exposed to extreme conditions like high temperature or high levels of water are enriched in other type of amino acids called the delta amino acids. You may think, oh yeah, but so why is this important? Well, it could be really important, because if for example, you work at NASA and they ask you, hey, Jose, if you have all the money in the world and could make a space mission, where would you send it to? And I will go like, to an asteroid that looks like this type, CM2, CR2, because they contain all the amino acids that are made for life, right? So that's the that's a kind of information that will be helpful for us. I don't want to go to an asteroid that has been exposed to high levels of water, although it could be interesting because we have found a relationship between meteorites and the levels of water in the sample. So the more water it has, the more the left-handed amino acids are present. So remember when I told you that amino acids could be left and right, and that we are left-handed? Okay, so the meteorites, which have been extensively aqueously altered, which means they contain a lot of water, contains the highest levels of the left-handed amino acid versions, right? So in a way, again, they will ask you, where would you, send the probe to? I will go, yeah, to this asteroid that is not too hot, not too cold, and that contains a lot of alpha amino acids, but has seen somewhat extensive aqueous alteration because it may contain higher levels of enantiomeric, left-handed enantiomeric axis. Okay. These are key factors for origins of life and for preparing NASA or space missions. Now, not only amino acids are important for life, of course, but that's not the only question that we have in the lab. We also want to know how these molecules are made in outer space and how these molecules could have, gotten their enantiomeric access in this case. Why are amino acids, enantiomeric enriched in meteorites? You see, this is an example of an amino acid called isovalene. Isovalene has been found it has been found that the left-handed version of isovalene in some meteorites, like Orgale, which contain a lot of water, is higher. So now the question that I have, okay, if we look at an analogous molecule like sec-butylamine, which is just like this amino acid, but it doesn't have the acid part, will we see the same enantiomeric axis? And the answer that we found is that no. We didn't find enantiomeric axis on this analogous molecule called amines. And that's quite surprising, actually, because if they were synthesized, we thought that the aliphatic chain here in gray would be synthesized to the same processes somewhere in space, and therefore it could be inherited, that the enantiomeric axis could be inherited. But we didn't see that. And so, lucky for me, someone in Germany was working on not the same project, but essentially the same question, doing different chemistry. So they were synthesizing amino acids using what it's called the circularly polarized light. This is a type of energy that would combine molecules under polarized light. And they found that if they use, for example, left-handed polarized light, they could enhance the production of one of the left-handed amino acids over the right-handed amino acids. And that's pretty cool, right? So because they could explain, hey, when there's no amino acids, but there's only gases like CO2, CO, water, ammonia, and they are combined into this like energetic location with polarized light, can synthesize and break symmetry. And that's neat, but breaking symmetry is not enough because we've seen higher enantiomeric excesses. And so that suggests that the amplification process that is needed would happen in the asteroid through the water that the asteroid contains, in a process that it could be like a recrystallization. And so molecules could wash off, like dissolve and rinse, dissolve and reprecipitate, dissolve and reprecipitate. And in that process, you could be enhancing the production, or you can be amplifying an initial enantiomeric excess that was broken in an interstellar location. So the theory goes like, you make a molecule in outer space, you break the symmetry of it like in a very small percentage, then these molecules gets incorporated into the asteroid, which is processed by water, which enhances, amplifies the enantiomeric excess. And then that enantiomeric excess, that asteroid hits the Earth, seeds the enantiomeric excess, and then life blooms using that enantio-rich molecule. Because, by the way, it's easier to put left, 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 left combined, as opposed to putting left, right, right, left, 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 left. You know, that's hard. So it's easier just to make one or the other, not a mix of them, right? Thermodynamics. And this same person did the same type of analysis, but with amines. So with the structurally analogous molecule. And they found that the amines were not supposed to break in symmetry. They were not supposed to have any amplification processes because their solubility, their physical chemical properties are very different from that amino acids. And so my experimental setup and results match to these theoretical investigations. And so, yeah, it was actually pretty good. All right, so let's rewind so that you don't get too lost. Organics are in outer space. They can be incorporated into asteroids, comets, and different planets, and but we can't get them in the form of meteorites, and we can analyze them as the most pristine natural samples that exist. They contain different molecules, including amino acids, which are key to the life on Earth, and homochirality, that is also, you know, what defines life. And studying other molecules is very important because they can give you clues about what processes could have formed all these key ingredients. Okay. But all that is not the end of the story. It's actually a tiny piece of the story because, and this is a challenge that the next generation has. We don't know what was the initial molecular abundance of certain compounds that we see in meteorites, right? So if I take a meteorite to the lab today and I analyze it, find the compounds, I quantify them, that's great. But that doesn't tell me if that amount is similar to the amount that was there, I don't know, 2 billion years ago, right? Or when the solar system was forming. We assume it's the same, but we know that there have been processing. So we know that it's not the same, right? And so we need to solve that problem. And also, our solar system is very fractionated, right? It went through the compounds in the different bodies have been destroyed, resynthesized, and destroyed again. Who knows? And so that all needs to that all have changed the molecular distribution, the isotopic values, even the enantiomeric excesses. So that's something that we still trying to understand, that we're not asking ourselves really strongly or like sufficiently. So yeah, those are areas of work that we are currently you know, we're seeing in the lab. But because I work at NASA, I can not only do meteorite analysis. I wish I could, but I can't. I need to work on space missions. And so space missions, specifically sample return missions, are really nice, are opportunities, you know, to analyze samples that cannot be accessed quite easily. The most famous ones are, of course, the Apollo missions. We brought a lot of like moon material to the Earth. And as a matter of fact, we're still analyzing that only recently. So it's like a time capsule, no? Other missions like the Stardust, which brought tiny grains of a comet to our planet, to our laboratory. and we were able to detect amino acids and other compounds, measure their chemical distributions and isotopic compositions. And more recently, the OSIRIS-REx mission, which is bringing a portion of an asteroid to my hands. So let me tell you about it. The OSIRIS-REx mission has a really funny acronym, but for a reason. It will tell us about the origins of the solar system and the origins of life potentially in our planet. It did a lot of analysis, spectral analysis of an asteroid. And it also did a survey of what resources could be used from the asteroid. Meaning, is it like covering diamonds, or is it covered in water because water is more valuable? And also, it had a component of security, but because in about 112 years, it has a small chance to impact the Earth. And so the orbit of this asteroid, the analysis of its orbit could change over time, and we could be better prepared than the dinosaurs if that happens. Although Venus is not that big, it's not big enough to be a like dinosaur-like event. But anyways, but okay, in any case, it's really exciting because it has a regolith explorer component, which means that the OSIRIS-REx will bring dust and material from the surface of the asteroid to the Earth. In 2016, it was launched, and about 2 years later, got to its the spacecraft reached the asteroid Bennu, and it stayed analyzing the surface for 2 more years. And on October 20, 2020, it did what we call the touchdown maneuver. So OSIRIS-REx was equipped, is equipped with a robotic arm that would flush a bottle of nitrogen gas to expel rock and debris, regolith, from the surface of the asteroid. And this material will be trapped in something that is literally similar to an old car filter. That filter would be encapsulated and brought back to the Earth. This is actual footage of the day of the touchdown. We reached about 0.5 meter deep, so we are going to get extremely unaltered material from an asteroid. As you can imagine, the surface of the asteroid has been exposed to Earth vacuum and weathering and radiation. But because this is a funny story, because we didn't know that the asteroid itself was going to be so soft. We were preparing to touch a really hard surface, and that's why we had the nitrogen gas, ready to blow the surface. But we were actually quite lucky that we didn't lose the robotic arm, just got trapped in the asteroid itself. So in any case, it was also good because we think that it will allow us to collect much more material than initially planned, to the point that we noticed that we were losing particles as the because there were 18 minutes between us knowing what happened, so there was a delay there. But we didn't want to put this into safety in the capsule, because we need to know exactly how much material we collected, right? But what we found is, like, we had too much and it was coming out of the system. And when we wanted to put back the filter in the canister, there was so much material that it didn't really fit. So they had to, like, wiggle its way. And In any case, it's on its way back in September 24th of this year. It's going to land in Utah, and we're going to analyze it probably in 2024. But this is all nice. However, the Japanese Space Agency has done this twice already with different levels of success. They have been to two different asteroids. One of them is the Ryugu asteroid that they touched down on December 2020. So the samples are already on the Earth, and we are lucky enough to collaborate with JAXA. So we are doing analysis of those samples as well. And yeah, so this is the age of the sample return missions. And we are happy to work and implement new methods for the use, for the analysis of these samples. It's better. Well, in some ways, it's always better to have the sample on the Earth, because you can do many analyses as opposed to those that you could do with a lander or in outer space itself. Just think of the Curiosity rover versus like bringing a piece of Mars to the lab, right? I'm part of this team for the soluble organic material analysis team.